Lechi lach to a land that I will show you. Lech lecha to a place you do not know. Lechi lach on your journey I will bless.
I want to thank you for inviting me to talk in this forum to these wonderful people. My lecture is titled Our Genetic Material and the Future of Medicine. I would like to dedicate my lecture to Susan and Elliot Winograd. So Susan passed away in 2018. Elliot passed away in 2016. He was part of your forum. You can see the certificate on the left-hand side. Elliot volunteered twice to come to Israel to volunteer in the IDF. In 2011 and in 2006, Susan and Elliot lived in Rockville Center, New York, and like a good Jewish story, it starts in Russia. About 130 years ago, our family decided to board a ship to New York, and the other part boarded a ship to Israel or Palestine then. And since then, I think we are discussing who's the lucky one, the one that took the boat to New York or the one that took the boat to Israel. Susan is my mother's second cousin, and we are in good contact with their children, Liz and Michael. We lived in Boston, where I spent some time at MIT, and I lived there with my family, with my wife and child, in 2014 till 2008. And at the time, Susan and Elliot were our closest family, our only family in the US, so we used to spend our Thanksgiving there and our Jewish holidays. So our genetic material and the future of medicine. In the future, would you prefer the clinicians, the clinical team on the right-hand side to take care of you, or the person, the so-called person on the left-hand side, the robot? Actually, when I asked my 10-year-old son, he immediately said, of course I want the robot to take care of me. But we'll see about that. It'll probably be a combination of both, and I'll tell you about it in this lecture. Imagine how wonderful it would be if we could predict the future. In fact, we actually can. We wake up in the morning and we take this supercomputer out of our pocket or next to our bed, and we ask it, should I take an umbrella today? Is it going to rain? Or which route I should take in order to avoid traffic jams. So we can predict the future. And this is based on advanced technologies, computer power. On the right-hand side, you can see 1 terabyte held in the fingers of the person. And on the left-hand side, you can see 5 megabytes, an IBM computer uploaded in the airport in New York. So the gap or the leap between the left-hand side and the right-hand side is about compression of 2,100 times more. And this leap was generated in about 60 years. Now imagine if we wake up in the morning and we ask this phone or supercomputer of ours, which drug should we take in order to avoid future diseases? Or maybe even, what should I eat in order to increase my memory? Or what sports I should take in order to feel happy? This will be possible based on not only computer technologies, but other technologies. This is advancements in our ability to read DNA. On the left-hand side, you can see a DNA sequencing or reading machine from about 30 years ago. This is the first automatic sequencing machine. It's called the Sanger sequencing machine, the first generation of machines that were able to read DNA automatically. On the right-hand side, you can see the second generation, the one that we use today. Now, the gap from the left machine to the right machine, from the first generation to the second generation, is 1 billion times more. We can read 1 billion times more in a leap of about 30 years, much more than the leap in computer science or advance in computers. That means that if we look at Moore's law, which claims that computer power increases every 18 months, and on the same graph, we will plot the cost or the ability to read one human DNA from beginning to end, you can see that the DNA wins by far, and the gap increases from year to year. This means that we could read much more DNA based on advanced technologies. Life expectancy in the past 200 years has increased from an average of 35 to an average of 80. If you go back hundreds of years or thousands of years, it would still be about 35. So the huge leap occurred only about 200 years ago. If we look at the population size on planet Earth, up till about 200 years ago, it was less than 1 billion, and today it's 8 billion. Now again, if we go back hundreds or thousands of years back, it was only a few million. So the question is, with so many people on planet Earth and increased life expectancy, how can we maintain the quality of life? How could we treat people not only to extend their life, but to extend their quality of life? And the answer is by adapting new technologies and assimilating them rapidly in the population. Now let's look at adaptation of various technologies. You can see here that again, in the past 10 and 20 years, there's a great increase in adapting new technologies. You can see the driverless cars, you can see the mobile phones and microprocessors. Just by comparing or looking how long did it take to get 50 million users, the landline or the phone, it took 50 years. For the credit card, a piece of plastic, 30 years. Mobile phones, 12 years. And we all know that now that if you have a really cool app on your phone, you could do that in one day. You could get 50 million users in one day. So we are adapting new technologies rapidly, and we are assimilating them very fast. But applying new data into the clinic takes longer. It's not enough to collect the data. You have to transform it into relevant information. And this information has to be translated into knowledge. And the knowledge should be led all the way to the clinic. So data has to undergo various filters before it reaches the clinic. And instead of dealing with sick care, we really want to take care of healthy people. It's a critical point. We don't want to be only reacting to sick people who come to the hospital, but we really want to take a stand and be proactive. Identify the diseases as early as possible. That would be a major leap in ability to deal with many of these diseases. In my lecture, I'm going to talk about the connection between biology and the digital world, how biology and life becomes digital or is becoming digital in the past few years, and also how the digital world is becoming more humanized, how a digital world really becomes human. Let's start from basics. So a building is built out of bricks, and our body is built out of cells. You can see it represented here by the hexagon. So we have many, many cells in our body, all attached to each other. Many cells form tissues. A group of tissues will form organs. A few organs will form a system, and all the systems in the body will form the entire body. And it works well. I'm sitting here, I'm giving the lecture. You're sitting at home, you're listening to the lecture. All these cells working together works terrifically. Now you can see here one human cell represented by the hexagon, and every cell in the center has the nucleus. If we increase the size, and if we could take a tweezer and take any cell from our body, from the tips of my fingers, from my mouth, from any organ in my body, any tissue, and we could open it with tweezers and take the nucleus out, we could pull the DNA out, the genetic material, the book of life. It's one tiny string all wrapped up inside the nucleus of all our cells. All our cells have exactly the same DNA. And this tiny string is actually very long. It's 1.8 meters, extremely thin, all wrapped up inside the nucleus in all our cells. That means that I'm actually sitting here and the cells are shearing off my hands, and DNA is shearing off my hands as well here. On my computer, you're sitting at your home, the cup you're holding, the pen you're holding, the glasses on your eyes, on your head, everything around you has some cells and have some DNA. That means that if we could take it, we could read the entire DNA of you or of me, if it's DNA around me, and we could read it from beginning to end and really understand how to build an entire human being from beginning to end, just by reading the DNA from any of your cells in your body. So here you can see a human body, a cute human body. And if we look under a microscope, actually the cells are not hexagons. Each one has a different shape. So the cells inside our GI tract look like fat cells with oil in them. The cells in our brain look like a ball with long extensions. The cells in our muscles look like rubber bands. The cells inside our blood look like small balls. And the cells on our skin look like stretched nylon. So all our cells look differently, but basically they are the same. They have the surrounding wrapping of them, they have the nucleus, and all the cells have exactly the same DNA. This DNA is the code of life, how to build an entire organism, just like you. So in conception, what happens in conception? Everybody started in exactly the same way. Our mother met our father, one thing led to the other, and then the sperm met the oocyte. During fertilization, half of the DNA from the father joins half of the DNA from the mother and forms one cell, one zygote. And you can see it here on the top right-hand side. We have one cell, half of the DNA contributed from the father, half of the DNA from the mother. This cell divides into two cells. Now, just before cell division, the DNA, the entire DNA, copies itself from beginning to end. And then one copy goes to the left, the other copy goes to the right. Now we have two cells. Two cells divide into four cells, 4 into 8, 8 into 6, 16. Every time just before cell division, the DNA would copy itself and split the copies between one cell and the other. Therefore, during a process which is called development and differentiation, you can see that when you have about 1 million cells, it doesn't look like a ball any longer. It has a head and it has a tail. This is a differentiation process. So every part in the developing embryo reads different regions of the DNA, and these different regions will tell it how to develop the head, the brain, and different organs. Now, with every division, you have the same DNA. That means that the embryo, the fetus, the baby, and also a human being will have all the DNA exactly the same in all the cells. Of course, there are some exceptions during cancer where the DNA changes or changes its sequence, and then it's a different DNA. But in a healthy human being, all the DNA is exactly the same. And all life forms are built from cells, just like I showed you, and all of them have DNA inside of them. That means that all the animals, all the plants, even bacteria or yeast are built out of cells, and inside they have the genetic code. If you read the genetic code, you will understand the book of life. And this is how it looks like. This is the code of the DNA. So code on a computer is based on zeros and ones. The code in the DNA is based on 4 letters A, G, T, and C. It's based on their chemical composition. So the exact machines that I showed you before, the first generation of sequencers and the second generation of sequencers, take a sequence of DNA, chop it up to pieces, and read the chemical content. After reading the chemical content, it actually writes it down, records it, and you could read the entire DNA from beginning to end. Actually, it's a special month and a special year, February 2021. Exactly 20 years ago, the first human genome was read from beginning to end. One person, one DNA, all the letters from beginning to end were read and published both in the US and in England. This was a huge project. Now, reading one DNA of one person from beginning to end, all the letters, took more than 10 years, and it cost more than $1 billion. It's a huge sum to read one DNA. 3 billion letters, 3 billion nucleotides, 3 billion bases of the DNA were read. And now there's the first puzzle. And now when you have the first puzzle, you could read additional ones. However, with such a cost and so many years, obviously it wasn't affordable and it wouldn't be reachable to many people. So the US government took it seriously and decided to generate incentives for many companies to reduce the cost, to develop technologies. And that led to the development of the second generation sequencers, just like the one that I showed you. And in 2007, while I was still at MIT, the first machines arrived. Big machines, but amazing machines. They actually were able to reduce the cost of reading one human DNA from beginning to end from $1 billion to $1,000. And it doesn't take 10 years, it takes 1 week. Actually, it was a gradual effect. It didn't happen in one day, but eventually this is the cost and this is the time it takes today. So it's wonderful. It means that we could read many of the DNA around us, and this is the key to understanding the development and the DNA, the genetic code of a healthy individual, and what happens when something goes wrong and when there's mutations and disease develop. So, how is biology becoming digital? How are we taking the life around us and turning it into a code? Before I tell you about it, I want to show you my lab. These are the wonderful students in my lab. Half of them perform computational experiments in bioinformatics, and half of them do wet work in the lab. Some of the experiments I will show you and tell you about were carried out by some of the students in the lab. How do we identify a genetic disease? So we work with many physicians and geneticists in Israel, and often they come to us with a small child, a girl or a boy, with developmental delays at the age of 2 or 3, for example. Now we take a swab from the mouth and we collect cells. We extract the DNA from the cells, and then after extracting the DNA, we place it on one of the DNA sequencers. We read the DNA from beginning to end, and then we scan it using computational power, and we try to identify the exact mutation or change in the DNA that might cause the severe phenotype or the external effects, the clinical effects. Often it's not easy. You have to run along the DNA 3 billion nucleotides and identify one letter that has changed. Sometimes it's a bunch of letters, or it's a complex disease with multiple changes. But in other times it would be 1 point mutations. And if we find the point mutations which is different to thousands of children or adults that we sequenced, we pull it out and we ask, what change does it cause? What might it lead to, which is also a computational process to try to understand what are the severe consequences of this particular change. And after we find the change in the DNA, it's a digital change now. We found it in the sequence, in the DNA sequence itself. We compare it to the databases. We sit with a physician and try to understand whether this particular change is known, has been seen before, has it led to any disease, are there any healthy individuals, children with this mutation, and what are the consequences? If this is a unique mutation, de novo, totally new, we build a mouse model based on it, and try to see whether the phenotype, the external effects, the clinical effects of this mutation are also visible in an animal model. Now, many companies and institutions and governments understand the power of reading large amounts of DNA. And there's a project in the US, 1 million DNA sequences of veterans are on the way. Now, this is a wonderful project because every veteran or every recruit in the US Army have their blood drawn and frozen in the freezers. And also there's medical records throughout the years. So actually they could go straight to the medical records, look at the medical records, they could go and extract the DNA from the blood samples taken many, many years ago. This project is on the way. There's also a large project in the UK, 100K UK. There's a large project in Asia that's running on the various populations in Asia. There's a huge project in China as well, and we're trying to lead a project like this also in Israel due to the diversity, the ethnic diversity in the population of Israel. The country is young, 70 years old. The population came from many countries, and if we sequence various populations in Israel, we'll have a very rich background of genetic information of the various ethnicities. So biology is becoming digital, but what about the other end? How is digital becoming more humanized? So the answer is through a computational process or field called artificial intelligence. So in artificial intelligence, machines or computers usually mimic cognitive functions that humans associate with the human mind. So having a computer think like a human being. So for this, you usually require a lot of data and large computational power. And this has advanced in the past 10 years. Therefore, artificial intelligence in medical processes is being assimilated in the past few years. Many of these projects and ideas were developed in the US, and some of them also at MIT. I want to mention 3 advances in artificial intelligence, not from the medical world, that really show how a computer could think like a human being. 10 years ago, the IBM Watson computer managed to win the Jeopardy challenge. I'm sure you're all familiar of the Jeopardy TV show, and they proved that based on this complex challenge of Jeopardy, the computer was able to think in advance just like humans do. And if you know this TV show, you know that the commentator would usually give the answer and ask the participants to give the question. And often they answer the question even before he finishes reading the answer. So you have to be very quick and you have to think intuitively like a human and not like a computer. There were major arguments and discussions whether this is real artificial intelligence. The final conclusion was that yes, it is. The computer was not connected to the internet. This is data that the computer read in books, in data, in text, and was able to assimilate and then to pull it in the right moment and to win this challenge. Another breakthrough in artificial intelligence was based on Baidu's image analysis. Baidu is the Chinese Google, and their artificial intelligence computer, supercomputer, was able to segregate pictures based on categories better than humans can. So here you can see the separation between a cat and a dog. It's not always that simple. And the human error was about 5%. The computer error was smaller than that. So they proved that computers are able to identify the image and segregate it into categories better than a human. Also, this is based on large data and large computational power. And in 2017, Google brought up a computer, generated a computer and an algorithm based on artificial intelligence that won the world champion in the Go game. Go is an extremely complex game. Go is more complex than chess. Like chess is more complex than backgammon. So in Go is like an entire war, and chess is like a small battle. And in Go, you deceive your opponent by playing some of your tools in one side of the game, and on the other side, you deceive it in another way. So this is associated with human traits, and after the computer has won the world champion, the Korean world champion, he decided to resign. Actually, the first computer, the AlphaGo, learned the game, and it took it many days to learn how to perfect it. The AlphaGo 0 had only 4 hours to play itself the game. It understood the rules, it played itself, it built its own strategy, and only then it went to play against the world champion, and he won the world champion. Now, a few weeks ago, luckily, Google decided to take their DeepMind computer, the one that played AlphaGo 0, and to generate an AlphaFold. AlphaFold will take internal cellular structures called proteins and to fold them properly. And if it folds them properly, this is the basis of identifying the structures within the cell and possibly generating novel and new drugs that can fit into various pockets into the proteins. So what we hope is that artificial intelligence would now be directed more towards medical applications or cellular applications, molecular applications, and assist scientists in their projects and not only categorize different photos online or on our phone. Now, how can we connect all of them together? How can we connect the biology to the digital world and the digital world to the humanized world? How can we bring it into the medical field? This is one example. This is a startup company from Israel called Viz.ai. I'm very proud to present it here. This is one of my former students from the lab. He was actually a postdoc at Stanford in statistics, and he suffered from a small stroke. And while he was lying in a hospital in Stanford and waiting for the physician to come and diagnose his CT images, he was thinking to himself, every minute matters. Every second matters. I'm losing my brain cells and nobody's coming to observe me, to test me, or to diagnose me. So luckily he was fine. He came back to Israel in 2016 and he established a startup company that automatically analyzes CT images and can segregate them to a stroke and non-stroke. And in the stroke, it could segregate them to high severity and low severity. He established the company in 2016. He got FDA approval 2 years later, and it's assimilated in 400 hospitals in the US now. So when someone arrives to the hospital and undergoes a CT with a diagnosis of potential stroke, the data streams to the physician's phones, it's analyzed there, and the severe cases pop up on top immediately. So it segregates severe cases from non-severe cases. It's extremely important, obviously, to save brain cells of the real strokes. It saves times, it saves money, and it saves lives eventually. This is an incredible technology, and this is based on artificial intelligence, teaching the computer to segregate between the various cases. Last year, there was a major publication on the ability of AI, or artificial intelligence, to diagnose breast cancer. So the system, the algorithm, was fed by 100,000 trained cases. These are US cases and UK cases. And then it was tested on 30,000 new cases. Now, as a comparison to the algorithm, an experienced, highly experienced radiologists from the US and the UK looked at it by eye and segregated it as well. Eventually, the computer-based AI system had 10% less false negatives in the analysis, and less 5% false positives. That the segregation by the artificial intelligence was much more accurate, and it's not far-fetched that in the near future we'll see the computers analyzing the mammographies rather than experienced radiologists. And also in the pathology work, in the pathology field, where pathologists have to sit hours over hours and segregate the slides and mark them for potential tumors, tumor tissues versus normal tissues. There are many startups in this field. There are many labs, university labs working on it. Some of these companies are also in Israel, and they assist physicians in the clinic to segregate tumor cells from normal cells in these slides. Now, how do we use artificial intelligence? So our lab developed an algorithm that can take two parents and a child, a diseased child, or could be also a non-diseased, a potentially diseased child. We sequence the DNA of the parents as the child. We crunch the data, we look at the potential mutations, and after that, we bring out an ordered list of potential mutations and rank their severity. This is a startup company that was spun up from my lab. It's actually an established company. It's located in Framingham, Massachusetts. It's called Variantics, and it serves the US market. So if you want to undergo now whole genome sequencing, you want to read your entire DNA from beginning to end for cardiovascular diseases, for potential cancer, for cancer mutations, for pharmacogenomics, which drug to take based on mutation, even amniocentesis during pregnancy, the US market is catered by Variantics. It's CLIA approved, and it's also HMO reimbursed in the US. And what are the future experiments that we're carrying out in our lab that are based on artificial intelligence, or generating biology into digital data and then taking the digital data and asking the computer to come to conclusions like a human would come to? What we're doing now is we're collecting blood samples of various types of cancers. So we have 4 types of cancers, cancer patients. We collect the blood samples and teach our system what the snippet or the small DNA fragments in these patients look like. And after we teach them what they look like, this is how the blood of this particular cancer looks like. This is how the blood of this particular cancer looks like. And there are differences between the two of them. We give them unknown, we give our system, we feed our system unknown, we feed our system DNA from unknown cases. And we were able to segregate the type of cancer based on a simple blood test, which can identify the snippet DNA released from cancer cells, which are different from these 4 types of cancers. In another case, we fed our system, our AI system, DNA that was retrieved from stool of colorectal cancer patients and controls. Our system read the DNA, built an algorithm for itself to separate the 2 samples apart. This is how the DNA from a stool of colorectal cancer patients, and this is how normal patients looks like, non-diseased individuals. And after the system has learned to segregate between the 2, we took unknown samples. We didn't tell the system what each sample is. We allowed the system to segregate the samples, and we have a very good separation in 90% of the cases between diseased and non-diseased. There's still a lot of development to take place in these projects. However, it's extremely encouraging, and we believe that eventually, using artificial intelligence, the system would be able to identify minute amounts of DNA, even in stool samples, and identify at a very early stage whether this is a diseased individual or a non-diseased individual. In another experiment, we fed our algorithm DNA of a fetus and DNA of the mother. Now, every pregnant woman at week 10 has some floating DNA of the embryo in her blood. So if we take a blood test and we read the DNA of the mother, nine portions would come from the mother and 1 portion would come from the embryo or the fetus. We taught our system how the minute amount of fetus DNA looks like, and we showed our system how the mother DNA looks like. And in our experiment, after teaching the system, we drew a blood from a pregnant woman at week 10, and we fed it into our system. The system read mostly the mother's DNA, and in some part also the fetus DNA. After a certain time point of reading enough DNA, we had an entire bag that the system told us this is 99% fetus DNA. Now we took this DNA, we rebuilt it like a puzzle, and we were actually able to read the entire DNA of a fetus from beginning to end at week 10 to 12 by a simple blood test drawn at week 10. This is a first AI-based system that can recapitulate the entire DNA of an embryo at such an early age without any risk for the pregnancy, like in amniocentesis, for example. This is extremely exciting, and we're trying now to establish a startup company based on this idea as well. So we talked about the genetic material and the future of medicine. And I asked you, would you like the clinical team on the right-hand side to take care of you in the future? Or possibly the robots on the left-hand side to take care of you? And I think the answer is that it's probably a combination of both. We will not give up on the physician coming and showing empathy to the patient, looking in the eyes of the patient, confronting them, comforting them, and telling them their diagnosis. But behind all these physicians, we certainly believe that there are going to be major computational power based on artificial intelligence. The biology will become digital, and the digital will be analyzed in a human way by a computer, but the data will be conveyed to the physician, and the physician will approach the patients, and they will talk to them, and they will convey the message directly to the patient. If you want to hear some more of my lectures, there's 2 TEDx talks of mine online, and thank you.
Professor Shomron, thank you so much. It was amazing. It was amazing, and well, just thank you. And before we turn to questions, I would like to ask you if you could possibly say something about the vaccines. You know, the most, this is the hottest topic right now we're all involved in. So we will appreciate your point of view on the topic.
So my first advice is go get vaccinated. So as you know, in Israel, 3 million people have been vaccinated already. 90% of the 60-year-olds and above had 2 vaccinations. It's important. So the idea is that this is a new vaccination. The vaccination is based on, instead of taking and weakening a live virus, like most of the vaccinations today, they're taking an internal part of the virus called an RNA. Even better than that, instead of taking the RNA from inside the virus, they generated a synthetic one, but it's identical to the RNA sequence inside the virus. Now they inject it into your shoulder. It goes inside the cells in your shoulder and it hijacks some of your cells. It generates the RNA and the protein inside your cells. Now it's exposed on the muscle cells in your shoulder, and your body undergoes stress and says, oh no, we're under an attack. Obviously, it's not a real attack. It's just the cover protein of the virus based on an RNA. Your body elicits an immune response. The first one is a small one. 3 weeks later, when you go and get your second vaccination, this is 10 times the magnitude is 10 times higher in terms of antibodies. And now you have antibodies in your body prepared for the real virus. For example, in Ichilov, in Soraski Medical Center at Tel Aviv, there are about 400 patients in critical condition this week. Only 1 or 2 of them had a vaccination. Most of them did not get vaccinated, even young patients. That means that the vaccination works. Now, this is an amazing time because in 1 year, the companies, and many of these companies obviously are in the US, were able to generate a vaccination and to ship it, I want to say all over the world, but looks like most of the shipping was to Israel. So the idea is that when it was sequenced in December 2019 in China, it was sent to the CDC, to the Center of Disease Control in the US, even before the first virus landed in the US. The CDC looked at the sequence and they immediately understood that it could be a major challenge to health organizations. Therefore, they suggested the companies to start generating a vaccination. In January and February, they planned it. It was possible based on the second generation sequencers that I showed you and identifying the exact sites in the RNA were based on AI and artificial intelligence. So that's the second technology. And the third technology was writing down the RNA sequence and generating the molecule. It's extremely incredible that this was done in 1 year. And again, we're in February, 1 year after the first virus landed in the US. I would say anybody who wanted to get vaccinated in Israel is vaccinated. I know the situation is different in the US. I have friends in California that said that they will receive it in the summer. I have friends in Boston that have received, and in New York as well, that have received the vaccination. But anyone in Israel that wants the vaccination received it. This is based on a revolution in the past 20 years. Reading of the human DNA, sequencing machines, AI that can understand the sequence, computational biology, and generating the vaccine, which is actually the easiest part of them all.
Okay. Thank you so much. That was regarding the vaccine, guys. So now let's turn to the questions. I will start reading the questions for you, Professor Shomron, and then, ladies and gentlemen, if you have any further questions, so you're welcome to write them down as usual. So the question by Jay, are you familiar with Yuval Harari book, Homo Deus, that discusses much of this subject? Yes?
Yes, it's an excellent book. Yuval Noah Harari has excellent books on the history of humankind.
Okay, it's written Yukul, and I didn't. Okay, Yuval Noah Harari. Yes, sure. Okay, yeah. Okay. Definitely.
Let's see additional questions. I also saw on the news that Israel developed medications that have cured 30 coronavirus patients. Yes, that's correct. So Professor Nadir Arber from Tel Aviv University, who I work with, worked on CD24, and they have an exosomes that surround their molecules. Inhalation really prevents severe cases. Now, I want to tell you that treating the disease is as important as vaccinations because everybody in Israel is vaccinated against one strain. If we have a mischievous second strain arrive to Israel, all the 3 million vaccinations are non-effective. Therefore, it's extremely important to develop medications as well. And by using these medications, you can eradicate or treat the coronavirus like a chronic disease, which is obviously much better. Do you foresee completely genetically engineered plants, animals, people, or even extinct species? Are there any laws protecting privacy of person's DNA from insurance companies, employers? Okay, so in Israel, there's a privacy law against using the DNA against you. in the US, I have friends that told me that if they should do life insurance before they read the DNA, so they won't, have to check the box in some of the insurance companies, have you read your DNA? Engineered plants, yes, completely. I want to tell you that by engineering plants, we are saving the world. There is not enough plants, there is not enough agriculture areas to plant the plants and to take the crops and generate food out of them. There's a few wonderful companies in Israel that do genetics on agriculture, and they increase the products and the fruits of many of the food that we eat. It's extremely important. Imagine a tree that would use the same amount of water and fertilizes and would give twice the amount of fruit. I believe that this will save the world, and we see these things as well. I know there's many issues with genetically modified organisms and plants. I personally don't think it's dangerous. I personally think it's going to save the world from hunger. How long do you think it will take before the majority of our population will be vaccinated? If you're talking about the US, it's up to your new Mr. President. At least I would say you have a new one now. His plans are probably different from the previous one. In Israel, everybody who wanted to get vaccinated got vaccinated. I received my second vaccination 1 month ago, okay? I'm vaccinated twice. That means I got the first vaccination 2 months ago already. Can you please explain why you can still pass COVID after you have vaccinations? Yes, you're vaccinated in your shoulder. Your blood is protected. It has antibodies. However, you carry the virus in your nose, in your mouth, and in your upper respiratory tract. You should treat these as well, and only by that you will be able to prevent passing the virus onwards. However, 2 studies that came out of Israel this week show that if you're vaccinated twice, you carry 50% less virus load. Okay, 50% less virus load if you're vaccinated. I have heard that it is better to have a reaction to the vaccination. Is this true? Not that I know of. What progress has been made in the treatment of multiple myeloma? I'm not quite familiar with that. I mean, I do work with physicians that work on this type of cancer, but what we usually do is we sequence the DNA, we give them back the data. Great information. I'm always impressed with the work and listening to your research. Oh, this is my third cousin, Elizabeth, Liz.
Well, she is right there. We all agree.
And I also have a personal note here. I won't mention the name, but this is my name, and I live in Tel Aviv. I was the program coordinator for Sarel from 2001 to 2019. I remember Susan and Elliot from their program here. It is so nice that you dedicated your lecture to them. The first time, this is another person, the first time I saw the cell phone in use, my ordinary people, was in my, soldiers taking their girlfriends to the bus. Okay, this is the personal stories. We live in fascinating times. Thank you so much for groundbreaking and sharing your data. Okay, these are the
Professor
questions.
Shomron, there is another question, if you could answer this one by Valerie.
Are there any over-the-counter medicines people shouldn't take for a time after getting vaccinated? Not that I know of. But you should ask the pharmacist.
Okay, great. Well, ladies and gentlemen, any further questions?
Yes. Yes, a question regarding the level of antibodies after the second injection. Is there a method to determine whether or not your T cells and all that stuff is high or low or medium?
Yes. You could take a sample of your blood and test it. I heard, for example, today someone was discussing in one of our groups. Someone felt terribly bad after the first vaccination, and they mentioned that the side effects, are headaches and nausea, and they're still suffering from it. So one of the physicians suggested to take a blood sample and to understand whether there's a good antibody effect, maybe an extreme antibody effect, maybe they wouldn't need the second vaccination as well.
Wow.
So yes, you can measure the amount of antibodies in the blood.
Could I ask a question?
Go ahead.
Yes.
This is not related to this. What happens to the role of the pathologist in the hospital with the computer taking over?
Okay, so the world is changing, okay? I remember I was in the US, I come to the US often when I can. I saw autonomous cars driving in Manhattan, a few years ago. Now, what's going to happen to the taxi drivers? We'll probably have less taxi drivers. What's going to happen to other professions? They'll just have to shift to another profession. Now, I'm not saying we wouldn't need pathologists. I'm saying they could use the computational system maybe to scan over more slides, maybe to look at the second opinion, maybe to speak with their patients instead of analyzing the slides all the time. Maybe they could look at their patient and not look at the microscope all the time. I'm sure they can find what to do. Pathology is really being advanced or assisted by AI technologies. And it's a wonderful fact because it means that there will be much less misdiagnosis when it's a computer. However, there's a major issue here. Who do you sue if the computer makes a mistake? Do you sue the computer, the person who bought the computer, the person who wrote the software? But these issues are in any AI system, okay? Also in autonomous cars. If an autonomous car would cause an accident or, God forbid, kill someone, who would you sue? Elon Musk?
Okay.
I have a question too. I'm Pamela. I'm the one who wrote you. Everybody on the screen knows that I was the program coordinator, so you could say my name. But with all of the people, I'm here in Israel, and I know in the US the same thing. There are so many people now who are the vaccine, with so many people that are not planning to get it. How is that going to affect the so-called herd immunity that that we want to have to protect everybody? What's going to happen?
That's a problem, but it would be mostly their problem, because if they will suffer from the virus, it might be detrimental. If you're vaccinated, it most probably would not be a detrimental effect. As I mentioned, 400 people are in critical care now in Ichilov Hospital. 2% of them, only 2% of them got vaccinated, and not even the second shot, okay? Only the first shot. That means that most people that get vaccinated are protected. If you're walking around people that are not vaccinated, they're still carriers. The thing is, you are also a carrier, but you can believe that at least your body is protected from a severe disease.
Okay, and also, do you think that in the sometime near future that kids are going to be able to, take the vaccine?
Okay, kids is more complex issue. First of all, I thought that if this disease would have hurt or damaged children, it would be extremely, extremely devastating. Unfortunately, much more than it is now. Okay, children, usually viruses harm the elderly and the very young. This virus is totally crazy in terms of symptoms, but it doesn't hurt children. Okay, it doesn't damage children. That's the advantage. So I would say it would take a long time before children would be vaccinated, if at all.
Thanks.
Can you tell us about any standard encyclopedias of DNA that you use as a reference?
So as I mentioned, 20 years ago, we had one DNA from beginning to end. And then, like anything in the US, someone took it very seriously and they said one DNA is not enough. So they developed these large machines that could read a lot. And straight away, the Broad Institute of MIT at Harvard and and Harvard initiated a project called 1000 Genome Project. They planned on reading immediately 1,000 additional individuals. This project went so quickly that it passed the 1,000 very rapidly, okay? Very rapidly, they changed it to 2,000 and then 3,000, and many projects followed it. So we compare now a sequence, a DNA that we read, to about 100,000 different DNAs sequenced. Again, this is led by the Broad Institute, okay? It's your tax money taking care of it. Now, if we compare it to 100,000 people, it's wonderful because we can understand if we find a mutation and it's present in the population, let's say 5% in the population, we can assume that it's not a detrimental mutation. If it's a severe mutation, we expect it to be extremely rare and actually even diluted out in the population. If we read a mutation and it looks like it's landed in an important site in the DNA, and we couldn't find any mutation like it in the Broad Institute 100,000 sequences, we can assume that this is a detrimental mutation, in particular if it lands in a protein that is responsible for a particular function in the cell.
Okay, well, since that we're done, unless...
Another question. By using mRNA, aren't we introducing a new element that didn't exist before, which might ultimately prove rather dangerous to the human genome looking forward?
So let's think about the virus entering our body. The virus enters our body. The virus has RNA, not only one mRNA, many RNAs. And these many RNAs are inside our cells and they could do whatever they want. By introducing one short synthetic RNA, I have it under control. And it's extremely unstable. That's the reason why they have to ship it in -70 degrees. If this wasn't a pandemic, nobody would approve a medication like that. Nobody wants to ship something in -70 degrees. Nobody wants to save something in -70 degrees. It's a big hassle. But because of the pandemic, they approved it. This is a non-stable molecule. It's non-stable outside. It's hardly stable in our body. And you would prefer a short, unstable, synthesized RNA rather than an entire virus with plenty of RNA, which is natural. Nature is much stronger than anything that we can synthesize or generate.
Yeah, Steve, I think the chances of the mRNA being incorporated into DNA is pretty slim in human cells. There's no, these enzymes aren't running around, you know, proliferating. So I think it's pretty slim. Plus, as Dr. Shomron said, the RNA is very unstable. That's why, as you say, it's got to be shipped in such, even though it's coated in a lipid particle, it still has to be, you know, refrigerated, stabilized in dry ice or less, or, you know, lower temperature like liquid nitrogen. So I think you're pretty safe.
And
Thank you, Jared.
It's an RNA, it's not a DNA, exactly. So it can't get incorporated into a DNA when it's an RNA molecule.
How about the effect on, thanks, Jerry. How about the effect on pets, dogs and cats?
Well, you know, when I went to school, we were taught that viruses don't jump species. But now after 100 years past, you know, my education, we now know that viruses jump species. And that's how a lot of these viruses who have arrived from China and other countries, you know, get into humans. So these spontaneous mutations, or whatever they are, you know, seem to allow for transmission, you know, from different species.
So like your previous president called it, the Chinese virus. So the Chinese virus is extremely mischievous. It intentionally generates mutations all the time. And 99.99999% of the cases, it's obsolete. It goes to the garbage bin. And in 1 in several billion cases, it works. Now, it worked in a tiny village in China, in one bat. There's millions of bats there. And that bat happened to find itself, to the market. That's what I heard. And then that bat was on a plate of someone in China. And that's the end of the story, or maybe the beginning.
This is why you should not eat bats. This is not kosher. There is a question by Valerie. How can we be protected from all information being used for negative reasons?
Yes, from AI information, from artificial intelligence. Well, first of all, let's hope that we would benefit from it more than we will suffer from this technology. This technology is used everywhere, again, in autonomous cars, in medicine. Actually, Elon Musk is one of the people who wants to restrict the use of AI. He believes that eventually it will take over the world. Some really clever computer will decide that it doesn't need us any longer, and it will try to get rid of us somehow. We have to be cautious, like with everything, every technology has a potential to harm, and we better use it in a proper way. We should have regulations, I could do anything. I could go and rob a bank, okay? I could do anything, but I don't, because I know it's illegal. I could drive really fast on the road, but I don't, because again, I know it's illegal. If you know what's illegal and you have people that abide the laws, then you hope that you're safe.
I see that you appreciate long-term trends, and I just wonder what your prognostication is, since the humans have eliminated smallpox and some cancers and so forth. How long do you think it's going to take the humans to get rid of this particular pestilence?
So I'm in favor of living peacefully with it. Let's turn it into the flu, okay? Something better than the flu, okay? The flu vaccines don't work as well as the COVID-19 vaccines. Let's turn it into something manageable. Let's turn cancer into something manageable, okay? Non-metastatic cancer. How about we stop metastasis and we leave the primary tumors where they are and don't even remove them? If we can control our diseases, let's learn to live with them. Have you read your own DNA? Why or why not? Okay, this is an excellent question. How about you Google my name and write, should you read your DNA? And there's a TEDx talk called, should you read your DNA? Okay, by me. And you'll get the answer. It's still the same answer.
I guess the answer is no, but I will read it. Okay. Well, Professor Shomron, thank you so much. You were such a great speaker, and you've answered all our questions. So thank you so much for it. And I hope you encourage some of the folks here about getting vaccines and then living with those who did not get the vaccines and living happy life in general. So thank you very much.
Thank you. And see you in Israel.